We talked about dynamic instability in the previous module. Dynamic instability is a process. It is much better if we can watch this process going on and then discuss it than just talk about it. But we had to talk about initially so you could understand what you would be looking at in the little movie clip that we'll be watching. Now I'm going to show you a still frame from this animation so I can explain to you what you would be looking at. Here we have a cell. In the middle, this dark stain you're seeing is the nucleus, and the bright spot on top of this nucleus is the centrosome. Centrosome is microtubule organizing center. We'll talk about centrosome later on. But here I would like to mention that microtubules are formed or nucleated at the centrosome. They are released from the centrosome, or they can stay attached. But microtubules form, they radiate out of the centrosome. So what you will see is these bright spots moving. What are these bright spots? These bright spots are tips of microtubules which are growing in the growing phase. How do we label these microtubules? Just a tip of these microtubules. They are specific proteins referred to as end-binding proteins. These proteins bind to the polymer when it has polymerized, your tubulin monomers, when they have polymerized, specifically at the plus end. How do they discern the plus end from the rest of the microtubule? Remember that we talked about microtubules being polar structures, and when the new subunits are added, beta tubulin has not initially has not hydrolyzed its GTP. So microtubules have this GTP cap on them, which basically means that beta tubulin, which is part of this polymer, is still bound to GTP and it has not hydrolyzed it. It has a different shape or structure which can be recognized by these end-binding proteins. When they bind it, they can light up the tip of the microtubule. This will only happen if the microtubule is growing. As soon microtubule stops growing, these proteins will fall off from the tip of the microtubule. So I'm going to go back to the previous slide. So you will see these white dots moving very rapidly, sort of like comets move. These are basically tips of microtubules which are elongating. In the next part of this movie, you will see microtubules. These microtubules have been the whole microtubule, not just the tip. The whole microtubule has been specifically tagged with molecules that can glow under certain light conditions. So you will see that these individual microtubules, they will shorten and then they will elongate. You will see this dynamic behavior of microtubules in real time. This movie was made somewhere between 20 and 30 frames per second. So you will be able to observe the dynamic behavior of microtubules inside the cell. So let's watch this animation. Here's the cell. Before we start the animation, I'm going to again point out this dark structure is the nucleus, and sitting on top of the nucleus, the bright spot is the centrosome. Please see these tips of microtubules. They are going to move very, very quickly. So please pay attention. I will stop the animation at the places where I have to describe something. So here we go. So these comets, as you can see, are moving very, very rapidly. These are the tips of microtubules which are adding subunits on. The glowing part again is specific proteins that bind the end of microtubules. You can see that how quick the polymerization of microtubules are, and also another point, how microtubules are nucleated from the centrosome and they're moving towards the periphery. So this is basically polymerization of microtubules. Now, this is a part of the process. Cell has extended its cytoplasm, and this is relatively a flatter structure. You can get a little better resolution in flatter structures. So again, you're going to watch these comets rushing towards the tip of this process, and see how they're pushing against the membrane, causing it to extend also. Here we have GFP tubulin. GFP basically is a special molecule. If you conjugate this molecule or attach this molecule to a protein, that protein glows under certain light conditions. In this case, we have attached this GFP to a tubulin molecule, and now entire microtubule array is lighted up. Again, when we focus on the periphery of the cell, you will be able to see microtubules in sharp contrast like here. Please look at the microtubules. These microtubules, specifically, they're going to grow and then shorten. So I'm going to start the animation again. It happens when this microtubule came out and then it went back in. This microtubule is extending and this is elongating, and some microtubules are elongating and others are depolymerizing. So I hope with this you will appreciate the dynamic nature of the microtubules. I hope you found these animations useful, and I hope they clarified some of the concepts that we had talked about verbally in the previous module.